Protein kinase is a PKA signaling pathway. Signaling molecule will go on to activate the G protein coupled receptor. The G protein coupled receptor will go on to activate GS, the G protein. The GS will go on to activate adenylyl cyclase. Adenylyl cyclase will go on to activate cyclic AMP. Cyclic AMP will inhibit ACAP, and ACAP is inhibiting PKA. And this is the picture that we have in the study guide, the cartoon picture that sort of depicts everything. So we're going to start taking this pathway apart piece by piece. And we're going to start with the part that is in the red rectangle. And this part is being shown in this picture, the activation of the G protein coupled receptor, where you have the G protein that is inactive with the alpha and the beta gamma subunits together and GDP bound to the alpha subunit. And upon activation of the G protein coupled receptor by binding of the signaling molecule, you have binding of the G protein with the activated G protein coupled receptor. And then you have the switch of the GDP for GTP in the G alpha subunit. And then the G protein is activated. And the G alpha subunit and the G beta gamma subunit will dissociate from each other and go on to activate separate pathways. So the G protein coupled receptor and the G protein are both tethered to the membrane. The G protein coupled receptor, of course, being a membrane protein, and the G protein is being tethered to the membrane by a lipid tail that is attached to the G alpha subunit and the G gamma subunit. The inactive G protein has a GDP pound at the G alpha subunit. Once the signaling molecule binds to the G protein coupled receptor, now the G protein coupled receptor can bind with the trimeric G protein or G protein. The activated G protein coupled receptor will act as a GEF, which will lead to the G alpha subunit exchanging the GDP for GTP. This exchange of GDP for GTP will lead to all three of these parties separating from each other, no longer being bound. The G beta gamma will separate from the G alpha subunit, and the G alpha subunit will of course separate from the G protein coupled receptor. Both the G alpha and the G beta gamma subunit will diffuse laterally on the cell membrane. The activated G alpha subunit and the activated G beta gamma subunit will then go on to activate intracellular pathways separately. It is important to note that the G beta gamma subunit is activated by just not being bound to the G alpha subunit. GS. GS is the G protein in the PKA pathway. It is just its name. Now we're going to look at the second part of the pathway. The activated G protein coupled receptor has activated GS, in this case the alpha subunit of GS, with its GTP bound, and now that alpha subunit of GS will go on to bind adenylyl cyclase. Upon binding adenylyl cyclase, the activated G alpha subunit of GS will go on to activate adenylyl cyclase, just by binding to it. So what does adenylyl cyclase do? Adenylyl cyclase is an enzyme that catalyzes the following reaction. ATP to cyclic AMP. This is the activation step, the on switch. The conversion of cyclic AMP to AMP is catalyzed by phosphodiesterase, or cyclic AMP phosphodiesterase. So cyclic AMP to AMP is catalyzed by this enzyme. This is the deactivation step, the off switch. And this is being shown in this picture here, where you can actually see the structure of ATP, cyclic AMP, and AMP. Now we're going to look at the last part of this pathway. Cyclic AMP is a second messenger like calcium. A kinase anchoring protein, ACAP, when active, will bind to PKA and inactivate it. ACAP subunits have cyclic AMP binding sites. Upon binding cyclic AMP, they will dissociate from PKA. And this is being shown in this picture here, where you have the inactive form of PKA. It is inactive because the catalytic subunits are bound to ACAP. Upon cyclic AMP binding to ACAP, what you will have is that ACAP will dissociate from those catalytic subunit, and the dissociation where the catalytic subunits are free of ACAP will lead to their activation. Inhibiting an inhibitor is an activation step, and that is being shown in this picture here. For example, for the PKA pathway, once the signaling molecule activates the G-protein coupled receptor, and the G-protein coupled receptor goes on to activate GS, and GS goes on to activate adenylyl cyclase, and then adenylyl cyclase will go on to activate cyclic AMP, noting that this activation step is increasing the intracellular or cytosolic concentration of cyclic AMP. This will lead to an inhibition of ACAP, and if you inhibit ACAP, then you're removing the inhibition on PKA, and that will lead to PKA activation. The PKA pathway is conserved in animal cells. Depending on cell type, the targets of PKA, a kinase, will differ. Just to make sure we understand, PKA is a kinase. It will phosphorylate substrates, downstream proteins in the pathway, and those downstream proteins will be different depending on the cell type. And that's being shown in this table here. For example, in the thyroid gland, you have the hormone TSH. This is the signaling molecule that will go on to activate the PKA pathway, which leads to an increase in cyclic AMP levels within the cell, and the response by the cell will be thyroid hormone synthesis and secretion. And you can see by looking at the different cell types that you have different signaling molecules which will activate different responses.